Go.
Why, hello there. My name is Professor Grumple McScufflebutt, PhD, Neuroscience. I have devoted my life towards the academic pursuit of the neuron. In order to understand this truly magnificent strand of cytosol, years of intensive study are required. You must be one with the neuron. You must feel the sodium and potassium ions coursing across your membrane. Did you cut? Or not. Neurons may seem difficult to understand and complex, but in actuality, their basic structure and function is relatively straightforward, not to mention fascinating. Neurons serve as a valuable illustrative example for many biological processes that you're expected to understand on the AP exam. So let's get started, shall we? In this video, we will cover the basic structure and function of neurons, membrane potential, diffusion and active transport in axon membranes, how an action potential is propagated along a neuron, resulting in the secretion of chemical signals into the synapse, the chemical signals neurons use, neurotransmitters, and neurons are amitotic. Okay, let's begin with the basic structure of a neuron. Neurons kind of look like little trees. They have branch-like extensions for receiving signals called dendrites, which feed into the soma, cell body, or, if you're feeling really fancy, the perikaryon. The soma contains all your standard cell stuff, including the nucleus. The axon is the elongated trunk-like structure down which impulses are propagated. Some areas of the axon are covered in a fatty insulating layer of lipids called the myelin sheath. Unmyelinated sections are called the nodes of Ranvier. The myelination of axons plays a big role in speed and insulation, which we will discuss later. The terminal buttons of the neuron contain vesicles of neurotransmitters. You may see this structure called synaptic terminals or axon terminals. When an impulse travels down a neuron, it eventually reaches the synaptic terminals, causing the secretion of chemical signals called neurotransmitters into the synapse, the junction between neurons. Note that the synapse is the junction, while the synaptic cleft refers to the actual space between the neurons. The neuron that secretes the neurotransmitters is called the presynaptic neuron, while the neuron receiving them is called the postsynaptic neuron. Are you ready? Now we're diving right into the function of neurons. Neuron function is often expressed with this graph, which models an electrical impulse arising in a neuron. Notice the x-axis represents time, and the y-axis represents membrane potential. Membrane potential may sound like a big scary term, but it just refers to voltage across a membrane, measured in millivolts. Due to differences in ion concentrations on the intracellular and extracellular sides of a membrane, one side may be more negative while the other side is more positive. This results in attraction across the membrane, the potential energy of which is the membrane potential. Our young neuron's story begins at resting potential. Now this may sound like our neuron buddy is just chilling out at the beach, but resting potential actually refers to the state a neuron is at when it's not transmitting an impulse. At resting potential, the membrane of our neuron pal is polarized and is at about negative 70 millivolts in membrane potential. The intracellular side is more negative and the extracellular side is more positive. This distribution of charge is due to the actions of our good old friend, the sodium-potassium ion pump. As you know, the sodium-potassium ion pump uses ATP to actively pump 3 sodium ions out of the cell for every 2 potassium ions it pumps in. Sodium and potassium ions both have a +1 charge, so by pumping out more positive ions than in, the sodium-potassium ion pump contributes to the polarized charge distribution. Also contributing is the fact that more potassium ion channels are open at this time than sodium ion channels, so potassium can flow along its concentration gradient to make the outside of the cell even more positive. This wonderful, oh so relaxing resting potential is disrupted when a signaling molecule, usually a neurotransmitter, binds to a ligand-gated sodium ion channel. When it binds to the protein channel, it causes a shape change or a conformational change in the protein, opening the channel and allowing sodium ions to flow along their electrochemical gradient into the cell. This is called depolarization because the inside of the cell is becoming less negative, so the membrane is less polarized. And here we have a beautiful example of a positive feedback mechanism. As sodium ions rush into the neuron and make the inside of the cell more positive, the resulting change in voltage causes voltage-gated sodium ion channels to open, causing even more sodium ions to enter the cell, making the inside even more positive, opening more voltage-gated ion channels. And, well, you get the idea. Now is a good time to mention some very important vocabulary. Once the membrane potential of the neuron reaches a certain threshold, represented as a dotted line on the graph, an action potential ensues, represented by the top point on the graph. It's important to note that action potentials are all or nothing, meaning that the neuron either fires, has an impulse leading to neurotransmitter secretion, or it doesn't. The impulses in neurons do not vary in intensity. That would be a graded potential. Anyways, let's get back to our membrane. That positive feedback loop doesn't go on for very long. Pretty soon, voltage-gated potassium ion channels open and sodium ion channels close, bringing the membrane potential down as potassium ions flow down their electrochemical gradient out of the cell. That little dip in the graph represents the undershot, during which sodium ion channels are closed, but more potassium ion channels stay open than at resting potential. This results in the membrane potential dropping below where it is at resting potential. Don't worry too much about this phase though, as it is more detail than what is needed for this course. Alright, so now our sodium-potassium ion pump is up and running again, and we're right back at the resting potential where we started. There and back again, a neuron's tale. Heh. So now we know all about how impulses are generated. But how do these impulses travel down an axon and cause the secretion of neurotransmitters into the synapse? To understand this, we are going to have to recall the myelin sheath and the nodes of Ranvier. The action potential jumps from node to node, traveling by a process called saltatory conduction. Since the areas the action potential affects are restricted to the nodes, the impulse is able to travel faster than it would if the depolarization effect had to happen at every point along the axon. When the depolarization effect of the action potential reaches the terminal buttons, it causes voltage-gated calcium ion channels to open and an influx of calcium into the neuron. The calcium ions promote the fusion of vesicles with the presynaptic membrane. Exocytosis of neurotransmitters occurs, and the chemical signals flood into the synapse, where they can bind to ligand-gated ion channels on the postsynaptic membrane. Neurotransmitters can have different functions and different effects on neurons. For example, depending on what receptor they bind to, the neurotransmitters can either bring the postsynaptic neuron closer to the threshold or farther away from the threshold. Neurotransmitters that open sodium or potassium ligand-gated ion channels have a depolarization effect and bring the postsynaptic neuron closer to its threshold for an action potential. This is called an excitatory postsynaptic potential, or EPSP for short. Likewise, neurotransmitters that open chlorine ion channels, chlorine ions have a -1 charge, hyperpolarize / make the inside of the cell more negative. They therefore bring the postsynaptic neuron farther away from its threshold and action potential, and are said to generate an inhibitory postsynaptic potential, or IPSP. Although the exam will not require you to know specific neurotransmitter functions, it is good to be familiar with some neurotransmitters so you will not be intimidated when you see them mentioned in AP questions. Serotonin and dopamine function in mood, with dopamine being especially notable for its function in reward pathways and addiction. GABA is inhibitory and glutamate is excitatory. Acetylcholine is the one most likely to be seen on the AP exam for its very important role in stimulating muscle contraction at the neuromuscular junction. Pop quiz time! Please pause the video to answer the questions. These are AP level questions designed to simulate the type of question you may see as a multiple choice question or FRQ. They require critical thinking and application of what you've learned. Number 1. Describe how the structure of a neuron relates to its function. Hint: think surface area to volume ratio. Number 2. The neurotoxin sarin prevents the removal of acetylcholine from postsynaptic ligand-gated sodium ion channels. How would this interfere with the functioning of neurons? Also, based on what you know about the function of acetylcholine, how do you think exposure to sarin would affect the human body as a whole? Okay, just one last thing before I go. Neurons are amitotic, meaning they don't divide and spend their time in the G0 stage. As you may recall, failure to pass the G1 checkpoint puts a cell in the more permanent G0 stage. Since neurons do not divide, damage to mature nervous tissue can be devastating. Brain tumors are usually due to unregulated growth of neuroglial cells, which are cells in the nervous system that support and nourish neurons, not neurons. Thanks for watching, guys. Good luck on the AP exam and have a great time in AP Biology. Let's get a yay for neurons! No? Okay, fine. Yay Biology. Peace out.